So in this unit about how we gather data, we've talked about the importance of randomness, and we've talked about how we go about doing a sample survey. Now we're looking at what it means to actually be an experiment and how is that different from an observational study. For an observational study, the main point is that a researcher is not going to make anybody pick any choice at all. They're just going to watch what naturally happens. And there's many ways to go about doing this. For example, you can actually start by randomly picking your sample and going off of data that was previously collected. For example, all right, I want to know about the students in my class. I want to know what were their choices for classes each year. Well, I could do a retrospective study where I select my students, the students that are in my class, and then I go looking at the data that was collected over them for their past history. I actually do that at the very beginning of every single year. I go back and I look at what is your math history, what is your AP history, and what's the likelihood that you're going to be successful in this class? Is there something we need to work at, or are you going to be one of the people I can rely on for help? Another thing that happens is instead of looking at past data, you might select your sample first, and then now that your sample is collected, go forward in time and watch what happens. That is a prospective study. So the main difference here, retrospective, you pick your sample and then you look back at previously gathered data. A prospective study, you pick your sample and you go forward in time collecting the data. Now, these are very important things that you can do. Observational studies tell us a lot. They let us see trends in our data, things that we might have related. We do observational studies to help us kind of look to see maybe things are independent. But there's a problem here. With an observational study, we are 100% not able to claim a causal relationship, a cause-effect relationship. That is absolutely crucial. And the reason why we have no cause and effect that we can examine here is because there is absolutely no form of control. The person doing the observational study is not actually impacting what's going on. You're not filtering out things that could be affecting your variables. You are just watching and seeing what happens. Now, if it is a cause and effect that we're looking to find, we can use a comparative experiment as long as that experiment has some sort of randomization built in. And that's because an experiment has a very specific design, and that design has elements of control that help us weed out other contributing factors. In an experiment, you're looking at hopefully one explanatory variable. You're really always focusing on one main idea, and that explanatory variable is called a factor, and that factor is something that can be manipulated to see what that does to your response variable. For example, I have done my own tiny bits of experiments in my classes in the past where I've said, okay, now we're going to do a flipped classroom. I'm controlling that one aspect. I want to see what happens when I change that one aspect of the class. Or what happens if I don't collect homework or I don't grade homework? What are the outcomes from that? Every year I do a little experiment. I pick one factor that I want to control. Homework, tests, quizzes, the lesson itself, study techniques, and I try to measure how that one factor affects the outcome. My outcome being my response variable. So for me, the response variable is going to be what are your grades. In an experiment, you have to have a couple of things. You have to be able to show that you are purposefully manipulating factor levels to create your treatments. For example, the factor levels that I've had, okay, let's do a 45-minute flipped classroom, a 20-minute flipped classroom, or no flipped classroom. And those are each my different treatments. Then we're going to randomly assign those subjects to those treatments. That's one place that I kind of fall short on because I don't have much choice. I have who's in my class this year. They get that treatment. Previous classes don't get that treatment. But we have the manipulation. Randomization helps make it good because it's going to be filtering out some of those biases. And the big thing here, if you're looking at this, we have three words. We have manipulate, that's what makes it an experiment. We have randomly, that's that guy. And then last but not least, we have comparative. So if we have a randomized comparative experiment, we should have those three things. Something that we're comparing our groups across the different treatment levels. For me, I compare what are the results of your AP scores and your test scores against how the lessons and how the class was designed for that year. And I try to figure out what is the best practice, what is going to have the best outcome. So going a little bit further into this, for a randomized comparative experiment, what we're really doing is we first have this level of manipulation. In order for something to be an experiment, the person conducting or the group conducting the experiment has to have some level of control. They are purposefully, actively, deliberately, they're choosing to change certain things within the study, changing the factors. That could be the level of a medication, could be the frequency, it could be how much light a plant gets, it could be where a house is situated on a street, but you are controlling certain aspects and looking to see how does that affect the outcome. But those aspects must be assigned by randomness because what we've looked at is that all of our samples can naturally vary from one to the other. And my samples themselves sometimes have hidden ideas, hidden things that lie under there that I might not know about. And by randomization, we try to balance that out. So if we have something extremely high, we hope to have something extremely low to balance it out. Hence the randomness being so important. We also absolutely have to compare, because if we don't have anything to compare it to, we have no baseline of data. We don't know, okay, has this changed? Has it improved? Has it gotten worse? Is it increasing? Is it decreasing? These are all comparative terms, which is why using the proper comparative terms in this class is so extremely important, because we're trying to figure out how do those changes affect my outcomes. So we got to identify who it is that's participating in this experiment. Remember, the who is the thing that you're measuring. The who in this case could be who our experimental units are. If we're talking about people, we typically call them experimental units, but they can also be called subjects or participants. Sometimes they like that term better because it feels more personal, it feels more humanized. It's not this cold, detached experimental units label. But if you're talking about mice, if you're talking about cars or computers or anything that's inanimate or we don't necessarily attribute emotions to, then those are obviously going to be your experimental units. And when we look at those experimental units, we have each factor. My factor might be, for example, trying to talk about somebody's depression. The factor might be a medication, it might be an exercise program, and diet. And in each of those factors, medication, exercise, and diet, we can have different levels. 50 milligrams of the medication, 100 milligrams of the medication. It could be different levels of exercise. You can exercise 3 days a week for 45 minutes each time, 4 days a week for a half hour, or maybe 3 days a week for an hour, and try to see how that affects. Those are your different levels. Same thing with diet. What type of a diet are you on? Are you on a completely self-chosen diet? Are you on a restrictive diet that keeps you from eating carbs? Are you on the zone diet, which looks for balanced amounts of carbs, protein, and fat? What are you eating? Each of those guys is called a level. So first we have our factors, which are the big overarching things that we think are going to cause a change in my experimental units, in my my outcomes. And then we have within each of those factors different levels that we're going to manipulate to try to figure out how it affects it exactly. And that combination of the end of all the different levels that are chosen for each factor, that is called my treatment. So one person's treatment for depression might be 50 milligrams of the drug, 4 days of exercise, 45 minutes a day, and on a restricted carbohydrate diet. Somebody else might have the treatment of being on 100 milligrams a day with 3 days a week of exercise and a balanced diet of carbs, protein, and fat. That is their personal treatment. And the treatments vary based on your factors. So when we do an experiment, these are extremely, extremely important. If you learn anything from this lesson, it needs to be the 4 principles of experimental design. Even if you just remember the main words for each step. That's important. Wink, wink. Nudge, nudge. The first principle of experimental design is control. Control allows us to kind of identify where sources of variation might be other than the factors and try to eliminate those as much as possible, or at least trying to make my groups as similar as possible across all of my treatments. Because if I know that one of my big things is that I have a person who is extremely sensitive to caffeine, I'm not going to measure their anxiety levels if they are having coffee in the morning. That's going to change things quite a bit. So instead, I might want to choose people that have the same types of reaction to external stimulants, so that's not factoring into my study. So control is first. Second is randomized. And you'll notice we started off this unit with randomization because it is such a key, crucial aspect of everything that we do in gathering data. Randomization, the whole purpose of this, is the fact that it should equalize the effects of things that we don't know or can't control. Now the problem is it doesn't completely eliminate those sources, but what it should do is over time, it should kind of like spread those differences out so that we're not blinded by them when we're trying to figure out what the outcome is. We control what we can, we randomize to take care of the things that we can't control, and without randomization, you absolutely cannot make any type of conclusions. That is true with surveys, sampling, it's true with experiments. You cannot draw a conclusion if you handpicked everybody because of a certain reason or another. You have to have randomization. After randomization, we have this idea of replication. So we control what we can, we randomize what we can't control, we always replicate our experiment, and the reason why we do that is 1 time might be a fluke. 2 times, okay, you're starting to see a little bit. 3, 4, 10, 20 times, and you're really starting to see a pattern. So the replication here is basically talking about 2 different aspects. First, replicating within a sample itself so that we have multiple people or multiple experimental units receiving the same type of treatment, but then at the same time, that entire experiment should be replicated as well. So sample sizes come into play initially, where we want to make sure that we have enough receiving each of the treatments that we can see that we have that variability spread out over different things. We can kind of get a better idea of what's going on, but then as well that we get to repeat the sample sizes again and again and again. So we have control what you can, randomize what you can't control, replicate to, and the last step is to block. And this is one of the hardest ideas to kind of wrap your brains around out of everything that we have here. The purpose of blocking is it says, okay, I know that there are certain types of characteristics that can affect my outcome. So what we do is we take any group of individuals or of our experimental units, and we say, okay, these have this type of characteristic that I think will affect the outcome, these don't. Or these have characteristic A and these have characteristic B. And so I separate them into blocks. It's almost like stratifying when we're talking about a survey, but blocking is what we do in an experiment. You group by common traits. The purpose of that is to reduce the variability within each block. So blocking gives us that halfway point between being completely random, where there may be a number of different things that are affecting my outcome, and so that's difficult to measure what's actually causing the final thing that we're measuring, my response variable, and full-on control, where we control absolutely every step of the process, including the selection. Because again, without any form of randomization, your whole process is wasted. So blocking is that nice little halfway point that will get me where I need to be. So here is an example of a diagram of an experiment. Experiments are really helpful when we want to kind of describe what's happening. The saying goes, a picture is worth 1,000 words, and that's exactly the case here. So we will start with a diagram to outline what the procedure is of my experiment. And you can kind of tell by looking at this. We start with a group. So we have one group, my population out here, and then from my population, I'm doing random assignment. If you don't say it, it didn't occur. So you must state when your randomization occurs. And you have to have that somewhere on your diagram. And for this one, I'm taking my group and I'm randomly assigning them to 3 different groups. I'm taking I guess we're surveying houses here, and I'm aligning them to 3 different groups. And then what I'm going to do is within each group, they get some sort of treatment. So this is treatment 1, treatment 2, and treatment 3. These are the things that I'm changing. So for one of them, my treatment has to do with what's going on with their meters. The other one is what's going on with the chart of their output, and then something about a control, which we will talk about later. And then what we have to do is within my group, you have to state what are you comparing, which groups are you comparing it against. So for example, we have group 1, group 2, and group 3. They're all getting compared. And what are you comparing? Okay. So this allows you to see that we have some main points here. You need to talk about the random allocation of your subjects, what are the treatments, and what are you ultimately comparing, across which groups, and what is your response variable. Now you might not specifically outline group 1, group 2, group 3, but you would have to tell me how many elements received each treatment. Now when we have blocking, things are a little bit different. We start with my main population. So this is my blocked design. We start with our main population over here. And then with this, we are basically, this is my blocking. We're blocking by gender. And then within each gender, we have random assignment. So there's my statement of randomization. I don't have it over here because I'm not randomly assigning people to be men and women. I know they're men and women. I'm grouping them by that. That's my block. And then within each of those groups, I'm going to random assign them to different groups which receive different treatments. In this case, they're going to be watching TV ads. And you do not need to state this. You do not need to state that it's group 1, group 2, and group 3, as long as you tell me what the treatment that each group receives. And usually it's nice to know the number that got each of those treatments, the number that you start with, and the number that goes to each of your treatments. And the thing that's very important is once we've blocked our subjects, never the two shall meet again. I'm only going to compare a reaction or response variable within each block. I am not going to compare the outcomes from the men to the women because I think that gender itself has a main effect on my outcome, so I'm not comparing those. Please understand again, we only compare within our block. We do not compare across blocks. All of this is about looking for differences between groups, whether they're control groups, whether they're blocked groups. You're looking for a difference. Now, it gets a little gray sometimes. How big of a difference does there need to be? Well, we hit this term that you're going to see over and over and over again, statistically significant. And that means that we're going to say, okay, what are the likelihood that I get these results if there is no difference? And that's usually if it happens less than 5% of the time. That's our go-to level of significance. That's where we say, oh yes, there's a big difference between these. And we are going to be getting very, very, very closely into that with our next unit where we actually start running tests. Now, when we say that we have a statistically significant difference, that means that my results, if the original was true, if the fact there was no difference, then the likelihood of seeing the results that you get are so slim that we're saying that original couldn't be true. My results are so out there, so crazy, that something else has to be going on. There has to be some element within there that's causing this outcome. And this is what we're looking for when we're talking about an experiment. Do you think your outcome is by chance, or is it attributed to your factors, your levels, your treatments? All right. So kind of summing this all up, when we're talking about an experiment and a sample, basically both the times what we're trying to do is gather data. But we have to have that our data is randomized in order to be able to get to that wonderful place of unbiased data. If my data is biased, it doesn't really do me any good at all. So we randomize to get unbiased data. But there are many different ways to do this. It has many different purposes. Some of them are similar from randomly selecting samples for a survey. So for a sample survey, you're going to randomly select your participants. But when you're doing an experiment, you don't always have that luxury. You can't make a person participate in an experiment. So experiments are usually done with volunteers. So it's kind of like how we had that voluntary response where people went to the survey to participate. For an experiment, people have to go to the experiment to participate. Now that in and of itself can cause difficulties because just like with a voluntary response survey, just the fact that the participants are choosing to participate, that can affect our outcomes. But that's why we randomly assign the treatments to our volunteers to reduce bias. So again, when we're talking about this, when we're trying to design an experiment, your first goal is to control what you can. So if you know there are certain characteristics or certain things that are going to affect your outcome, you control what you can. But you can't control everything, and you have to have some level of randomization. So we randomize what we can't control, and that's oftentimes how our treatments get assigned. We will first off control what type of people are allowed into the groups, or maybe I will block by factors that I know might affect my outcome, and then I randomize how the treatments are assigned. Once I do that, it's always good to replicate your process over and over again so that you know that your results aren't a matter of a fluke or something that could have happened. There might have been a mistake in the process, an error in the process. But then our last goal is to block if we can, because again, the blocking is our way of taking care of the things that we know to be effective. Now we will go over this more in depth in class, but you want to make note of what we said. This is a great opportunity for you guys to fill out your flip books and be prepared for class. Thank you very much.